Hello, everybody. Today we are going to talk about energy and metabolism. Now, you've probably heard the word metabolism throughout your life. It's usually in reference to somebody's weight or somebody, saying, "Oh, I have a super high metabolism. I can eat anything I want." Or more so in my case, my metabolism is starting to slow as I age, and therefore I have to watch what I eat. So, what does that mean? Well, basically, your metabolism, is the rate at which your body is completing all these chemical reactions. And the faster your metabolism is, basically, the more calories you're burning, and therefore, the more calories you can kind of eat. So, we're going to break that down and kind of talk about what is energy, and then how is energy and metabolism, how are they related, and what exactly, how do our bodies run on energy, and how do we then, perform metabolic reactions within our body. Okay, having said that, here we go. Energy is the ability to do work or bring about a change. And there are two types of energy. There are kinetic and potential energy. Kinetic energy is energy that is in motion, and it is mechanical, therefore it is movement. So the example here is water going over a waterfall. You could harness that energy and use it somewhere else, right? Potential energy is energy that is stored, or chemical energy. The energy you get from eating a carrot. You eat that carrot, your body then breaks down the nutrients in that carrot and gives your body energy. Okay? So kinetic and potential are our two types of energy. There are two laws of thermodynamics. And I know that's a scary word, but it's pretty basic if you just kind of break it down. So the first law is that energy is always conserved, meaning energy cannot be created or destroyed. It rather just changes from one form to another. So photosynthesizing leaves capture the solar energy. They then use that energy to form carbohydrates along with the energy and carbon dioxide and water, and they basically make that into food for themselves, right? And that's their energy. So all you really need to know is that the first law of thermodynamics basically is telling us energy is not created or destroyed. It is basically just changing formats. The second law is the law of entropy. And that means when energy is changed from one form to another, there is usually a little bit of a loss of energy that is available then to do work. So basically, these transformations of energy types are not 100% efficient. And usually, we get a loss of energy that is in the form of heat. So, the processes occurring in our cells are energy transformations. Every process in cells increases the total entropy in the universe. Therefore, less work is available to do useful work. Meaning, glucose tends to break apart into carbon dioxide and water over time. Glucose is more organized and less stable than its breakdown products, meaning carbon dioxide and water. Just like if you have a neat room, it is much more organized, but maybe less stable than a messy room. Meaning, if you have all these boxes stacked in a neat column, they are much more likely to tip over than if they are already laying on the floor. And it requires you to take energy to put these boxes back into that tower and become neat again, right? So those same principles can be applied to our bodies, to plants, to the usage of energy within our world. So we have organisms that are called producers, and they use energy to create organized structure in biological molecules. Producers are plants. Then we have organisms that consume those plants and use that stored or potential energy to therefore drive their processes. We are consumers. We have to take in food. We cannot live off of sunlight, right? We do not photosynthesize. So we have to eat that stored or potential energy to give us energy. Okay? And this is constantly going around and round, and we need that solar energy to start this whole process, meaning photosynthesis. Okay? So, that brings us to what is metabolism. Metabolism is the sum of all cellular chemical reactions taking place within a cell. And you have what are called reactants, which are the items that participate in the reaction, and you have products. And this is what is formed as a result of that reaction. Free energy is the amount of energy that is available to do work. There are two types of reactions, exergonic and endergonic. Exo usually stands for out, endo usually stands for in. So in an exergonic reaction, the products have less free energy than the reactants. So these exergonic reactions release energy, and they can happen at any time. Endergonic reactions mean that the products have more free energy than the reactants, and they require energy to be put in, and therefore they are not spontaneous, because energy has to be put into the system to drive that reaction forward. Our cells run on ATP. That's the energy of the cell. Adenosine triphosphate, ATP. It is a high energy compound that is used to drive our metabolic reactions. ATP is not stored by cells. So that means it constantly has to be generated from what we call ADP, adenosine diphosphate. ATP has 3 phosphates stuck onto it. Adenosine diphosphate only has 2. So we have to make ATP through cellular respiration, and then we break it down. Then we pull off that 3rd phosphate, and that's where we get our burst of energy. So it is a constant cycle of our bodies having ADP, us taking in a food source, us making ATP, and then us ripping that 3rd phosphate back off for the boost of energy. And on and on and on it goes. Okay? And that is how we use energy in our cells. So this process is called a coupled reaction. Energy is released by the exergonic reaction and is captured in ATP. ATP is then used to drive an endergonic reaction, which is what I just described. It goes on and on and on in a cycle. So, we also have things called enzymes. So reactions normally occur in a sequence, meaning you have a start point and you have a stop point. Okay? Products of an earlier reaction then become the reactants, also known as substrates, of a later reaction. And we just keep going on and on and on. When these reactions are linked, they form what is called a metabolic pathway. It begins with a particular reactant, proceeds through several intermediates, and terminates with a particular end product. In other words, the product of the first reaction is the substrate of the second, and so on, except for in the case of the letter G here in the example, because that is the final end product. So, as I said, you're going to start with your initial, let's say the A here, which is the reactant, or sometimes called the substrate. And this process is going to go through very methodically. It's going to go through these different sequences, and you're going to get B, which then goes into C, and C goes into D, and D goes into E, and E goes into F, and boom, G is where we end. And that is our final end product. Okay? And that whole sequence from A to G is called a metabolic pathway. Sometimes we need these special proteins that are called enzymes to drive these pathways forward. Enzymes are protein molecules that function as a catalyst. The reactants of an enzymatically catalyzed reaction are called substrates. Each enzyme accelerates a specific reaction. Each reaction in a metabolic pathway requires a unique and specific enzyme. That's important. Any old enzyme can't make a metabolic pathway go. It has to be the specific enzyme for that reaction. The end product will not be formed unless all enzymes in the pathway are present and functional. So now you've got your A through G, but it's showing that you need all these 6 enzymes to drive each of those reactions forward. Okay? And there is something called an enzyme substrate complex. So there is an active site of an enzyme, and it basically binds with the substrates. So it's what we call, as it says here on the bottom, an induced fit model. So the enzyme is induced to undergo a slight alteration to achieve optimal fit for these substrates. So basically, when the enzyme meets the substrate that it's supposed to work on, they join together, and it causes the active site to change shape. The shape change forces the substrates together and an initial bond formation. So the enzyme comes in, it kind of locks into the substrate, and it allows these reactions to take place. And again, enzymes are specific to their substrates. Degradation. Enzyme complexes work with a single substrate molecule. The substrate is broken apart into 2 product molecules, which are then released. Synthesis is when the enzyme complexes with 2 substrate molecules, and the substrates are then joined together and released as a single product. Degradation, breaking down. Synthesis, building. So enzymes can work in either direction. The enzyme is just driving the reaction forward. Doesn't matter if it's a building reaction or a degradation reaction. The enzyme is just there to make sure that this reaction proceeds. And that brings us to what's called the energy of activation. Molecules frequently do not react with one another unless they are activated in some way. Okay, so they have to be stimulated to go forward. Energy must be added to at least one reactant to initiate the reaction. And that energy is called the energy of activation. And that comes into play with enzymes. So the EA, the energy of activation, prevents molecules from spontaneously degrading in the cell. So the enzyme operation, what is it doing? Well, the enzyme is operating by lowering the energy of activation, meaning the enzyme comes in and lowers this energy of activation level so that the reaction can then proceed forward. If there was no enzyme, that energy of activation would be too high for that reaction to ever occur. So enzymes lower the energy of activation. This then triggers the reaction to go forward, to proceed. This influences the rate of reaction and is why enzymes are catalysts of chemical reactions. The enzymes are driving these reactions forward by lowering the energy of activation level, because without doing that, nothing would happen. So it is the enzyme that is the catalyst in this reaction. Here is a picture of what I just kind of explained, showing that when that energy of activation comes in, it allows the reaction to progress, as you see along the x-axis there. You start at the same point, you end at the same point, but basically the amount of free energy that you need is less because you have lowered the amount of energy of activation due to the use of the enzyme. See, the yellow line is when the enzyme is present. So it has lowered the amount of energy that needs to be put in to drive that process forward. The blue line is where it was originally with no enzyme. Very high. Couldn't happen. Okay? Pretty cool. What affects the speed of an enzyme? Well, there's a couple things. Substrate concentration, meaning how much substrate there is. Enzyme activity will increase with substrate concentration due to more frequent collisions between the substrate molecules and the enzyme, right? If there's a lot of substrate moving around, there's going to be a lot of reaction. Temperature. Enzyme activity can increase with temperature. Warmer temperatures cause more collisions between the enzyme and the substrate because molecules are usually moving around quicker when they are heated up. However, there is a spot there that becomes too much. If something is too hot, it will denature or destroy the enzyme. And we know this because proteins become denatured if they get too warm, right? And enzymes are just a specialized protein. So we see the same thing here happening as we see when a protein becomes denatured. If an enzyme is heated up too much, it will lose its ability to work because it will unravel just like any other protein. Here it's talking about an example, which is that Siamese cats have a mutation that causes enzymes to be active at cooler temperatures, and that's what gives them those special colorizations. Meaning, if you've ever seen a Siamese cat, some of them have like tipped paws and a tail, and they have their ears or their nose are a little bit different colored. I think they're adorable. I love cats. But anyways, it's because of enzymes that they get that cool tipping of coloration. And then we have pH, okay? Most enzymes work specifically at a certain pH level. Remember that pH level 1 through 14, 7 is neutral. Anything below 7 is acidic. Anything above is basic. So just like a protein, if a certain enzyme is made to work at, let's say, pH 9, which is basic, and you throw it into some type of solution that is pH 2, which is very acidic, it is probably going to shut that enzyme down. Okay? So main things: substrate concentration, temperature, pH. Factors affecting enzymatic speeds. So here are some extra things. Cells can regulate the presence and absence of an enzyme. Cells can regulate the concentration of an enzyme. And cells can activate or deactivate some enzymes. This is all very good to know. So basically, our cells are regulating the amount of enzymes, which is good, right? We don't want these enzymes just getting, just doing whatever they want. Everything has to be checked. So your cells are kind of running the show. There are things that are called enzyme cofactors, and these are specific molecules that are required to activate an enzyme. This could be FAD, NAD+, or NADP+. And those are the first two in cellular respiration, and they are the third in photosynthesis. So if you have ever studied cellular respiration or photosynthesis, you will see these things within reactions, and that is because they are working as cofactors to help activate enzymes so that those processes can proceed. There are also things called coenzymes, and they are non-protein organic molecules, such as vitamins. Okay, and they help regulate enzyme action as well. Then there is something called enzyme inhibition. And this is a reversible enzyme inhibition, meaning a substrate known as an inhibitor could bind to an enzyme and therefore decrease its activity. So there is something called competitive inhibition, and this is where the substrate and the inhibitor are both able to bind to an active site, so they compete for one another. So basically the inhibitor looks just like the substrate, and it can get in to the enzyme and there render it useless. Whereas usually you would want the substrate to come in and perform the correct action. And as it says here, basically it then as I said, this inhibitor is going to render the reaction useless because the product is only coming when the substrate is binding with the active site, not with the inhibitor. The inhibitor, hence the name, is inhibiting the reaction from happening. And then there is what is called the non-competitive inhibition, and this is when the inhibitor does not bind at the active site, but it binds somewhere else on the substrate and changes the shape of the substrate so that now the enzyme can't fit into the active site. It has rendered the active site, like it cannot pass into the active site. The active site is no longer good because it's been changed enough that the enzyme can't fit in. Because remember, they're specific to the substrate. So what you need to remember is the competitive inhibition is when an inhibitor is going to go right into that active site, therefore blocking it. Non-competitive is what we call allosteric, which means that something got in somewhere else in the substrate and has changed the active site configuration. Enzymes can lead to death. Enzyme inhibitors can lead to death, right? If these enzymes can't get in and can't do what they're supposed to do, we could have a problem. So materials that irreversibly inhibit an enzyme are known as poisons. So cyanide inhibits enzymes that are required for ATP production. Well, that means you're not making energy anymore. Cellular respiration is ceasing. Cellular respiration is how we live, humans live. Serine inhibits an enzyme located at the neuromuscular junction. This means how your nerves and muscles talk. And if your muscles can't talk to your nerves, it could end in paralysis, right? You're not going to have any muscle function. And warfarin inhibits an enzyme responsible for the blood clotting process, meaning you stop the ability to clot your blood, meaning you could basically internally bleed out. Those things are poisons, right? Hence the word. Okay, couple other things here. There are things known as oxidation reduction reactions, and they play a role in metabolism. I see we're missing an O in that metabolism. Anywho, electrons are always passing from one molecule to another. Oxidation is the loss of an electron, whereas reduction is the gain of an electron. And I know that might seem counterintuitive because you're saying, well, why would a reduction be a gain? Because electrons carry a negative charge, right? Think back to chemistry. Electrons are the negatively charged subatomic particles. So if you are reducing, you are gaining a negative charge. I know it's kind of tricky. It takes a minute to think about it. And this oxidation and reduction are taking place at the same time. So one molecule or atom accepts the electron given up by another, and it is just a constant cycle. So when we make salt, NaCl, sodium is oxidized, meaning it loses an electron, and chlorine is reduced, meaning it gains an electron. There's a little thing there to remember. Oil rig. Oxidation is loss, reduction is gain. So photosynthesis and cellular respiration are both metabolic processes. Photosynthesis is where you have carbon dioxide and water and solar energy making glucose, and the byproduct is oxygen. The plant lives off the glucose. It spits out the oxygen, which we then take in with food or glucose, and we perform cellular respiration and spit out carbon dioxide, water, and we have energy from the process. Boom, that plant then takes that carbon dioxide that we gave off as waste back into its starting one of its starting reactants. Hence why the world goes round and round. Photosynthesis, cellular respiration, photosynthesis, cellular respiration. This is why we should talk to our plants and stop cutting down the forests, right? We need plants, people. They're the only thing that makes oxygen. So keep that in mind. So this is just a little bit more information on that. And chloroplasts capture the solar energy and use it to convert water and carbon dioxide into a carbohydrate, which is glucose. Hydrogen atoms are transferred from water to carbon dioxide as the glucose forms. So back to that oxidation and reduction, carbon dioxide has been reduced and water has been oxidized. Okay, and the energy is the solar energy from the sun that is driving photosynthesis. On the flip of that, in the mitochondria, which is producing cellular respiration, we have the mitochondria, which is oxidizing carbohydrates and using the released energy to build that ATP, the energy of the cell. Cellular respiration consumes oxygen and produces carbon dioxide. It is exactly the opposite of photosynthesis. Here we have glucose being oxidized and oxygen being reduced. Okay? How cool, right? This cycle, it blows my mind every time I talk about it. It's amazing. The photosynthesis, the cellular respiration, and so on and so on. Okay, so that's where we're going to end. This is a constant cycle. It works out really, really well, amazingly, for the most part, because each cycle is giving up, or, their products are exactly what the other guy needs for its reactants. So it really just keeps going in this wonderful, beautiful biological metabolic pathway to sum it all up. So there we go. I know it's a lot of information, but obviously metabolism is a super important topic. It is driving life, if you want to put it that way. So I hope you enjoyed the lecture. Please let me know if you have any questions or concerns, and have a great metabolic day. Bye-bye.